Good afternoon. Welcome everyone to the EPA's Cum Risk Assessment 2013 webinar series. This webinar series is co-sponsored by EPA's Risk Assessment Forum, Cumulative Risk Assessment Technical Panel, and EPA's National Center for Environmental Research, and we're happy that everyone's able to join us today. My name is Devin Payne-Sturgis with EPA's National Center for Environmental Research. I am the Assistant Center Director for Human Health, and also with me on the line is my colleague Lawrence Martin with EPA's Risk Assessment Forum. And we are honored to have with us today a leading practitioner on cumulative risk assessment and ecology from EPA's National Center for Environmental Assessment, Dr. Glenn Suter. The first part of the webinar will last about 30 minutes, and once Glenn has presented, we will have approximately 30 minutes for questions and answers and discussion. And as usual, since there are many participants on the phone, and in order to ensure a successful webinar, all lines have been muted to minimize background noise. And I'm also going to ask my colleague Ashley to go over the ground rules.
While all lines have been centrally muted to reduce background noise, please also use the mute button on your phone if possible to avoid background noise, which can make it difficult for other participants to hear. If your phone does not have a mute button, it can be muted through the computer using the microphone button next to your name on the participants list on WebEx. Please note that muting using the WebEx participants list should only be necessary if you have accidentally clicked on your name in the WebEx participants box. All questions will be submitted using the chat feature in WebEx. These will be compiled and the presenter will address as many as possible following their presentation. To access the participants list and the chat feature in WebEx, please click on the green tab at the top of your screen. This will expand the box and you click on either the chat or participants, and a box will appear on the right-hand side of your screen. This webinar is currently being recorded and will be made available on the EPA NSER website.
Well, thank you, Ashley. Before we get started, Lawrence and I would like to say a few words about the purpose of these webinars. The purpose of this series is to share and discuss the progress of groundbreaking research on cumulative risk and other related activities, to develop and apply methods for assessing the combined health and/or ecological risks of multiple chemical, physical, and/or biological stressors with consideration of population vulnerability. The series will feature scientists from both inside and outside EPA. Assessing or evaluating the true reality of environmental impacts on human health is a major challenge for the agency, and from my perspective, there is no one approach to cumulative risk assessment. There really is a spectrum, and our purpose with each webinar is to explore an aspect of cumulative along that spectrum. Last month, we had a presentation by one of our STAR grantees on a novel statistical and analytical method to overcome a major challenge of data availability at the proper geographic resolution for community-based cumulative risk assessment. Today's presentation will be on integrating across human and ecosystem health outcomes for cumulative risk assessment. So now, Lawrence Martin will say a few words about the Risk Assessment Forum's efforts on cumulative risk and how today's webinar will help address important issues for the RAF's efforts. Lawrence?
Thanks, Devin. This is Lawrence Martin, in the EPA Office of Science Advisor. I staff the Risk Assessment Forum's Cumulative Risk Assessment Technical Panel, which is charged with the development of the guidelines for cumulative risk assessment. We have 5 writing teams at work now, drafting the guidelines, and we're making progress. These seminars continue to provide thoughtful insights to CRA methods that the writing team reflect upon. The RAF CRA Technical Panel published the framework for CRA in 2003. You can see the link at the bottom of the slide here, and it remains the seminal foundation document for our work. The Risk Assessment Forum is pleased to partner with NSER to bring these webinars of current cutting-edge thinking on CRA, and panel members listen to both the speakers and the dialogue following the presentations to gain insights to CRA methods, as well as to hear critiques and conceptual concerns. Next slide, please. So here's the technical panel membership, and while you're admiring the talent here, let me mention that the RAF published a request in the Federal Register on May 1st requesting information and citations on approaches and methods for planning, analysis, assessment, and characterization of cumulative risks to human populations and the environment. This notice solicits information and citations pertaining to approaches and methods that can be used to plan and conduct cumulative risk assessments. It requests that information be posted to the EPA public docket, and it's going to close in 37 days. To see the notice, Google Federal Register and cumulative risk, and it should be the first thing that pops up. Okay, so on behalf of the RAF CRA Technical Panel, we're really happy to welcome a member of the RAF Steering Committee, Dr. Glenn Suter, who chairs the RAF Ecological Oversight Committee. Glenn's been a leading thinker at EPA on how to better integrate human health and ecological assessment information. And this is a critical issue for CRA, and I'm very much looking forward to his presentation and the following discussion. So welcome to the CRA webinar. Glenn, yet for another 15 minutes of fame. Back to you, Devin.
That's great. And I'm really excited that the Risk Assessment Forum decided to do that Federal Register notice to solicit input, and I really encourage folks who are participating in this webinar today to check out that, that announcement and provide your input. It's really important for us. So Lawrence gave a little bit of introduction, but I'll give also a more formal introduction for Dr. Glenn Suter, who is our 11th speaker in our series. Glenn's presentation is entitled, Why and How to Integrate Your Assessment. Glenn is a science advisor in the EPA's National Center for Environmental Assessment in Cincinnati. He's also chair of the Risk Assessment Forum's Ecological Oversight Committee, as you just heard. He has a PhD in ecology from the University of California, Davis, and over 35 years of professional experience. He is the principal author of 3 texts in the field of ecological risk assessment, editor of 3 other books, and author of more than 200 other publications. His research experience includes development and application of methods for ecological risk assessment and ecological epidemiology, development of soil microcosm and fish toxicity tests, and environmental monitoring. So with that, I'd like to turn it over to Glenn.
Thank you. I hope everybody's seeing a nice blue EPA logo there with the title. What I'm going to be talking about, as you just heard, is integrating assessment. And the reason that we need to do that is summarized by this first slide. We have one world. We don't have access to alternative universes. We all, human and non-human alike, are exposed to the same environment and the same pollutants. And we humans, in dealing with those problems, need to make one decision, a permitting decision or a remedial decision, a policy decision, that will meet the needs of and protect humans, non-human organisms, ecosystem processes, and even economic entities like corporations and political entities. All of that needs to be integrated into our assessment and decision making. So by integration, I mean a lot of different things and use them in different contexts. The first one, and the one that the cumulative risk team has been dealing with primarily, is multiple stressful agents or pollutants. But there are also multiple media and routes of exposure that need to be considered, multiple endpoints, human health and eco-receptors, cancer and all other effects of stressors, different levels of organization. On the eco side, we deal with effects at organism, population, community, ecosystem, and even global levels. Multiple types of assessments, and I'll spend some time on that later. And risk to socioeconomic systems as well as humans and non-human systems, and that gets us into the arena of sustainability, which I'm sure most of you are familiar with. I believe that integration is imperative. Otherwise, your risk estimates are going to be incomplete if you're not looking at things in an integrated way. And more importantly, if scientists don't present a coherent and consistent assessment across those issues that I was describing, then decision makers often won't do the integration for you. They'll simply dismiss unfamiliar science and make a decision based on the parts that they understand. Stakeholders will be confused if you present a disjointed result for assessments, and will tend to not accept the results you present. And as a result, bad decisions will be made. So, with that introduction, I'll spend a bit of time talking about how, on the eco side, we deal with multiple agents and stressors. And we're dealing here not just with chemicals. Since we deal with the environment explicitly, ecologists have recognized that when we consider toxicology, we also need to include temperature, things like suspended sediment, physical habitat structure, nutrients, dissolved oxygen, and all the other aspects of the environment that impinge on organisms. And of course, humans are also influenced by habitat characteristics, although in different ways. So how do we handle multiples? Now, first, like the human health risk assessors, we tend to deal first by looking at the data on individual chemicals and other stressors and modeling using first exposure additivity and effects additivity models. And then if we're dealing with a complex case, we may use combined additivity, where we look at exposure additivity for chemicals or agents with a common mode of action, and aggregate up within those modes of action using effects additivity models. Modeling is common, but it's highly unreliable. It doesn't include environmental and chemical interactions, and it doesn't include toxicological interactions. It doesn't deal well with unmeasured chemicals for obvious reasons. It's not very applicable to non-chemical agents like temperature, and it doesn't deal well with the influence of the conditions under which the exposures to the multiple agents occur. So, recognizing those problems, sometime back the ecological risk community started saying that we should let the organisms tell us about the mixtures, and that means exposing them to the mixtures and not just to the single agents, and also not just exposing them to a defined mixture with a few components, but exposing them to the mixture that they experience in the environment. So we started with diluting aqueous effluents with receiving waters and developed tests for that, and starting in about 1980 with a set of standard methods for freshwater and saltwater. And you can see from the cover in the lower right that we're now on the 4th edition of the chronic toxicity test for effluents and receiving waters. So this is a well-developed and commonly used practice. We also have tests now for contaminated sediments, soils, and even waste materials. And this is just to give you an idea of what that looks like. This is a crew out collecting water from a stream to take back into the laboratory and perform tests. This is a diluter that will take that water or an effluent and dilute it down so that we can run a concentration series. And I included this because I was impressed recently by the need to do just the opposite in human health toxicology. In a recent study of the effects of disinfection byproduct mixtures in drinking water, it was necessary not to dilute the water but to concentrate it so that you could get it up to a level that might cause effects. So that gives an impression of where on the scale of toxicity human and non-human organisms are with respect to environmental mixtures. These are the two most common standard test organisms for freshwater fathead minnow and a crustacean zooplankton named Cereodaphnia dubia. And these are tests being run with those organisms. On the left there, the little wells contain Cereodaphnia dubia, which can, during the 7-day course of the test, actually go through a life cycle and produce neonates. And the fathead minnow larvae in the beakers on the right, the larval stages are the most sensitive stage, and which is convenient because also the smallest stage and fit in those nice little beakers. So we run these tests, and the next problem is if an effluent or receiving water or soil or whatever is toxic, what's causing the toxicity? And we've developed a set of techniques called toxicity identification evaluation that consist of tiered treatment and testing of the contaminated materials involving fractionation, removal of fractions, adding things back in, retesting, until you can identify which component of a mixture is causing the toxicity. A new approach that's being developed primarily in Europe is called toxicity profiling, and it consists of performing tests and then looking for characteristic biomarkers in the organisms, and from the array of biomarker responses they hope to be able to identify the causes of toxicity. Obviously, mixtures toxicity testing is a better way to go than single chemical toxicity testing if you have the mixture available and organisms that respond. But you still are testing a relatively small number of species, and often the most sensitive species isn't included, like this mayfly naiad that you see on the right is more sensitive than Cereodaphnia to a lot of inorganic contaminants. You may not have the life stages you need, like the spawning adult fish. You may not have the durations you need for chemicals that bioaccumulate over time. You don't have species interactions, and you don't have environment interactions. You're still in the laboratory. So another approach is to do biological surveys. You can do these for any sort of organisms, plants, wildlife, etc. This is a crew doing electrofishing in a stream, and this is basically equivalent to an epidemiologist walking down the street and tasing the people he meets and weighing and measuring them and examining them for pathologies. Advantages and disadvantages of field surveys are that both come from the fact that you're dealing with the real world. So you get some sensitivity because you're including all species and all life stages within a community that you're surveying. However, they can be insensitive because the effects we look at in the field are population and community level effects, which may be less sensitive than the organism level effects that we can see in the laboratory. And also, we have the same problem that human health epidemiologists have, in that the causation of field survey results can be obscure. So back in the 1990s, primarily, the EPA was encouraging the states to do a lot of biological survey work, particularly in streams. And as a result, the states were identifying thousands of impaired streams, but really had no idea what to do because they didn't know what the cause was. So the agency developed a method for determining causation in ecological systems called stressor identification, which was actually based on human health epidemiology from Hill's criteria, and then from Susser's expansion of Hill's criteria and his scoring system. And we expanded this into an expert system and called CADIS, the Causal Analysis Diagnosis Decision Information System, which you can find online. So if you've collected all this evidence, single chemical toxicological evidence, mixtures toxicological evidence, biological surveys, maybe even some field toxicity tests, you've got multiple lines of evidence, and what do you do with that? How do you make an inference when you've got disparate lines of evidence and it may not all be consistent? Well, we have two ways of dealing with that on the eco side. One is independent applicability, which is based on the idea that each type of evidence will miss some effects, but any detected effects in a well-conducted study are real. And therefore, you believe the effects you see, and you don't take quite so seriously the effects you don't see in other lines of evidence because of their limitations. The alternative approach is the weight of evidence approach. Each type of evidence is recognized as having strengths and weaknesses, and we weigh them based on those strengths and weaknesses and the quality of the evidence and its relevance to the case, and determine whether effects are real and whether effects are caused by a particular agent of concern. So how does all this ecological practice relate to what human health assessors do? Human health risk assessment has been more reliant on single chemical toxicology than ecological risk assessment. Toxicological studies of ambient mixtures are rare for good reasons. Human exposures tend to be much lower, the effects are more subtle, and the tests tend to be quite expensive. It may be possible to bypass this with techniques that are becoming available now, but that's the state of practice. Epidemiological results are used, but they're often treated as independent from the toxicological results rather than being weighed against each other or their relative applicability being determined. But there are some exceptions, and one excellent example that I saw recently is the dioxin reassessment that did a good job of treating epidemiological data and toxicological data in an integrated way. So there are methods that are being applied for treating multiple lines of evidence in human health risk assessment, but they're pretty much ad hoc, as in the dioxin assessment. The team pretty much developed their methods as they went. So given that, I want to tell you about 3 case studies. The first one is one that I was personally involved in. It's the Poplar Creek embayment on Watts Bar Reservoir in Tennessee, a Superfund site, contaminated by mercury, PCBs, and radionuclides. But the mercury and PCBs were really the big problem. And since mercury and PCBs are both bioaccumulative in aquatic systems, the main concern was for the high trophic level piscivorous consumers, fish-eating birds like that great blue heron, fish-eating mammals like the mink, and fish-eating humans like the guy from Frank's Car Wash. And I was lead on the ecological portion of this assessment, and we recognized that we had a difficult and potentially important problem here, so we also had a large and willing funder in the Department of Energy. So we did biological surveys. We looked at reproductive success of great blue heron rookery that was on the embayment, at kingfisher colonies, or not colonies, kingfisher burrows. And we did a mixtures toxicity test. We actually netted hundreds of pounds of fish and shipped them to Ann Arbor, Michigan to be fed to mink to look at the effects of consuming the mercury and PCB mixture on reproductive success. And in fact, we found that even diluted down the consuming fish from that embayment reduced number and weight of kits. The human health risk assessors at the same time were using standard human health risk assessment methods, looking at mercury toxicity and PCB toxicity independently, and using standard exposure models. And when we went to them with our results, which we thought would be of interest to them, they said, no, we can't use that, which caused all kinds of problems when we presented the results to the public, because they wanted to know why we were saying that there was no risk to humans when we'd shown that there were reproductive risks to the non-human organisms. And there was a lot of hand-waving and there was spontaneous explanation going on at the public meeting. So a little integration in advance would have been helpful in that case. Another example is the Coeur d'Alene River Basin in Idaho. It's part of the Rocky Mountain Mining District. Number of lead mines and other mines in the headwaters of the Coeur d'Alene River. And this is another Superfund site. As you can see in the lower right, the people were exposed to the mining waste during floods. The mine tailings came into the towns. But the wildlife and fish were even more exposed. We had massive die-offs almost every year of swans and ducks. And we had a large portion of the river, which should have been supporting the threatened bull trout, which you see in the upper right, in fact, didn't support the fish because of metals toxicity. The EPA decision on this site was based on strictly compliance with water quality criteria and with achieving soil cleanup levels. They didn't use the wildlife toxicology that was done, even though the Fish and Wildlife Service did run tests of the contaminated sediments in the lakes and showed that they did in fact kill waterfowl, and tests were run of the water and sediments and showed that they were toxic to aquatic life. And when the National Research Council reviewed this site as an example of how the agency was handling mining Superfund sites, they wrote a rather scathing report called Superfund and Mining Megasites, which is at the URL that you see on the slide here, and chastised the agency for not doing a good job of integrating human health and eco concerns and coming up with a remedial action that would take care of all the receptors. The last example is one that we're trying to get right now. The proposed Pebble mine in the Bristol Bay watershed of Alaska is considered by the people who live in the Bristol Bay, who are primarily Yupik, southern Eskimos, to be a threat to their livelihood because it's a threat to the salmon fishery. So we have here a case where the people involved have presented a request to the agency in terms of an integrated assessment problem. They want the agency to protect the salmon in order to protect them. So we've been engaged with the native communities there. The woman that you see in the red cap is over 100 years old. She speaks no English, and the woman next to her is translating, but we got testimony from her and from actually hundreds of other people in this village of Nustoyahook and other towns and villages in Alaska about what the salmon mean to them, how they relate to the salmon, what they believe the mine would do to the salmon and to them, and both in terms of their subsistence consumption, which is averages over 300 pounds of salmon per person per year, and in terms of their commercial livelihood, like the commercial drift net boat that you see in the upper right, which is owned by the Yupik gentleman that you see in the picture. So that's what I mean by integration, those three examples, but integration also can come into play across the environmental spectrum, registering pesticides, permitting outfalls, etc. And in all those cases, there are benefits in terms of efficiency. If we work together, there'll be less duplication of effort. Scientific quality, if the best science from all the scientists is used. Completeness, looking at unconventional and indirect exposures and effects that may not be part of the standard human health models. And the non-human organisms are good sentinels. They are the metaphorical canary in the coal mine because they're more exposed, they can be sampled, they're often more sensitive than humans. They're certainly, in many of these cases, more abundant, so there's more chance of exposure. And examples of chemicals that have had obvious effects on non-human organisms that are perhaps controversial in terms of how much effect they have on humans are DDT, dioxins, tributyl 10s, and estrogenic compounds. Efficiency, go into these issues in more detail. From shared information comes from the fact that the sources are common to both human health and eco. The physical chemical properties are common. The transport and fate processes are common. Often, the exposure processes are common to some extent, although humans don't respire water, for example. And there are often common modes and mechanisms of action across organisms. It seems obvious to me, but in fact, I've seen cases in which different approaches were used to extrapolate from rats to humans and rats to mink in the same assessment. Different models used to estimate aqueous concentrations on the human health and eco side, separate searches of the mammalian toxicity literature even, and different plant concentrations used in dietary models for humans and wildlife. And in particular, on one mercury-contaminated floodplain, on the eco side, we collected plant materials and analyzed them to determine plant uptake, and even planted a garden so that we could look at uptake dynamics, and included some vegetables that humans would consume. But in fact, the regulators determined that those data could not be used in the human health risk assessment. In fact, you had to use a standard uptake factor model where the concentration of plants was simply a standard uptake factor times the concentration in soils, ignoring the fact that the uptake was quite different for mercury at this site than in the standard studies. So, human health and ecological risk assessment, I would argue, should be more consistent. We should share a same or consistent scenario about what we believe will happen in the future, the same or at least consistent assumptions about issues like how exposure might occur, consistent spatial and temporal scales, comparable effect endpoints. For example, if I'm looking at reproductive decrement in river otters, you should probably be looking at reproductive effects in subsistence fishermen, so that we can give a consistent story to the people when we roll out the assessment. And perhaps the most difficult part, we should have common expressions of probability and uncertainty. This is sort of the heart of risk assessment, and it's the most difficult thing to convey. And it's even worse when the ecologists and the human health folks are using different statistical approaches and different expressions of results. And to do integrated assessment, we need an integrated framework. We need to be approaching risk assessment in fundamentally the same way. And back in 1998, WHO, the OECD, the European Union, and the US EPA created a working group to develop an integrated human health and ecological risk assessment framework and approach, and to apply it to some case studies. And that was completed some time back. It's the report's available on the WHO site that you see there, and in a series of papers in the journal Human and Ecological Risk Assessment. And here's the framework that we came up with. The integrated risk assessment framework is based largely on the ecological framework because it was clearly superior to what was available at the time on the human health side. And there was an emphasis in this group on the parallel processes of risk assessment, risk management, and stakeholder participation. The EPA's Risk Assessment Forum is now completing a draft human health risk assessment framework that you see here that is also based largely on the ecological risk assessment framework, which is also the basis for the cumulative risk assessment framework that some of you are familiar with from prior webinars. And the advantage of having this common framework is that we're speaking a more common languages. The steps in the process are the same: planning, scoping, problem formulation, an analysis phase, and a characterization phase, so that there's a better basis for collaboration between the human health and eco-assessment teams, and a more common structure to our products and a more common language when we're speaking to decision makers and the public. So I'm very encouraged by the fact that the human health risk assessment framework is shaping up in the way it is, and we're going to have this opportunity for integration. Prospects, I think, are also good because toxicology is becoming more mechanistic, which should encourage integration because there's a lot of commonality at molecular levels. But also, environmental science is becoming more holistic. It's becoming more place-based, dealing with larger scales, and if you're dealing with a real place, even if you're a traditional human health risk assessor, you've got to recognize that the environment is impinging on the people in ways other than presenting them a chemical at a time. And I believe that both of these trends can promote human health and ecological risk assessment integration, and that, I believe, will all be to the good. EPA has been moving towards integration. The Risk Assessment Forum's Cumulative Risk Assessment Framework and the current guidelines effort, which Lawrence referred to earlier, includes an integrated panel of human health and ecological scientists. The National Exposure Research Laboratory has a program producing integrated exposure models. The CREM, or agency-wide modeling team, has an integrated modeling initiative. And on the eco side, we have an ecosystem services research program, which has been going on for several years, trying to bring our ecological risk endpoints closer to the concerns of humans. But I believe that in general, the European Union is a little further along because their REACH legislation, which is their chemical safety legislation, mandates an integrated assessment be performed. And so they've been funding research on integrated risk assessment, the biggest of which was called No Miracle. The Europeans love those clever acronyms. And even the medical and veterinary community is involved to some extent through what's known as the One Health Initiative, trying to look at common approaches to human and animal health. But I want to emphasize what I don't want integration to be. It's certainly not diverting the ecologists to work on human health, as one EPA manager suggested some time back, that all the ecologists should be working on the ecology of disease vectors for human diseases. That didn't go over very well with the ecologists for obvious reasons. It's not even diverting them to assess human welfare, such as benefits of walk in the woods, or even risks to subsistence cultures like the people in the Bristol Bay. We've been doing those things. I've been working on the Bristol Bay assessment and meeting with the people there, but it's not what I'm trained to do. It's not really what I know how to do. And even with the help of University of Alaska anthropologists, I'm not sure that we're doing as good a job as we could if we had people who were actually trained to be assessors of environmental effects on human welfare. So rather than trying to push ecologists to being human welfare assessors, I think the human health risk assessors and the eco risk assessors need to meet in the middle, perhaps hire more social scientists and cover the whole range from purely ecological concerns through the human-environment interface to pure traditional cancer risk assessment. I'm going to talk to you just a bit about integrating types of assessments because I'm using up my time. Risk assessment isn't everything. We also need to do environmental epidemiology to determine what conditions are problematical and what the causes are. We need to do predictive assessments other than risk assessments like cost-benefit analyses. And we need to do outcome assessments to determine whether our decisions have the benefits that we hope for. But don't integrate for the sake of integration, and don't do it in a way that diminishes your effectiveness. but rather integrate in a way that better informs decisions and provides a coherent understanding of the consequences of alternative actions based on the best available science, whichever stovepipe it comes from. And I think we can do this best if we have an integrated worldview that hazardous agents enter ecosystems, and within ecosystems there are people, and broadly they interface with ecosystems to affect human welfare, and human health is a subset of that. And this Yupik people in Bristol Bay, through their interface with the environment, I think have that view of the world built into them from the very beginning, and they can serve as an example to the rest of us of how to interface humans in the environment. So thank you for your attention, and we'll entertain questions.
Yes, thank you so much, Glenn. We're very happy that you're able to join us today.